Hi, and welcome back to the EDGE tutorial series. This video will demonstrate use cases for the reference-based analysis module in EDGE. After this tutorial, users will know when to turn reference-based analysis on and how to select parameters for this tool. As a reminder, we are running two job submissions in parallel. The first uses E. coli FASTQ files found in the PublicData testData directory in EDGE. The second uses data from the SRA run SRR 2164314. Reference-based analysis is a useful tool for investigating samples of known composition. For instance, studying a pure bacterial culture. This module maps reads and contigs to references selected by the user to obtain coverage information and identify uncovered regions where sample reads or assembled contigs do not align to the reference. The output of this module provides information on variants, SNPs, and uncovered regions that do not align to the reference. Interactive visualization using JBrowse to display the references, align reads, and contigs are used for output. An example of that looks like here, what you see on the right. The bottom image is an example of zooming in to a particular location on the reference. Within the EDGE interface, the reference-based analysis tool must be turned on. The user then selects one or more reference genomes for comparative analysis. Users have the option of uploading their own reference genomes as FASTA files. Lastly, a user must select either Bowtie2 or BWA as the read aligner. Both aligners are going to yield similar results. The distinction is based upon user preference. Bowtie2 is selected as default. With this information, we're ready to set up this module in our EDGE demo. Let's begin with our E. coli sample. Scroll down to the reference-based analysis. Note that it's turned off. We want to switch that toggle on. Click on it to have the drop-down menu. We know this sample is a pure E. coli isolate. Let's select some E. coli genomes to compare it to. If you click on the list, type Escherichia and click Enter to see available options. Now let's select some of interest. We suggest that users select up to 10 relevant reference genomes. We won't select that many today. I'm going to select these three K12 strains. Go back up to the top and click X to save your selections. Since we do not know the composition of our metagenomic SRA fecal sample, running reference-based analysis doesn't make much sense up front. It may become useful in subsequent analyses after we have an idea of what is present in the sample. For now, we will leave this module turned off, which is default for this demo. We can double check by scrolling down to the reference-based module and seeing that it is off. Okay, that's it for this video. The next tutorial will introduce the taxonomy classification module.